Hi, this is Ritik, sir, your biology subject matter expert from memoneet. So, in this video, we will be concentrating the remaining part of the biotechnology and its processes, that is, the separation and isolation of DNA fragments. In the previous video, I have just summarized like how do you separate and the DNA fragments under gel. So, today we will discuss even in deeper to it than the PCR technique and how you're gonna get the desired DNA or whatever the material into more copies and its applications. Everything we will be discussing in detail. So, when coming to the separation and isolation of the DNA fragment, so one thing that is you are gonna choose a DNA, and with the help of the restriction endonuclease, so we will cut the DNA into a smaller fragments of DNA, then these fragments can be separated. So we have huge fragments of DNA here, right? So now these fragments has to be like separated. So this fragments will actually be separated by using what? Gel electrophoresis technique. So what is this gel electrophoresis? Is nothing but since we know that the DNA is negatively charged, molecules, so we can separate them, forcing them to move towards the positive charge, that is towards the anode, right? So under how an electric field through a medium, so or through a matrix. So the here the medium or the matrix that we are discussing, the most common used medium or matrix will be like agaros or agaros. Okay, so agaros or agar. So which is actually a polymer extracted from a seaweeds, right? So the DNA fragments separates according to their size. That means if they are very small in size, they'll move first, and if they are very huge in size, they'll move very slower. So the smaller the fragment sizes, the farther they move. As you can see here is in wells, you will load the sample, which consists of the DNA, and the largest size is here. So they all are, as we know, it's negative charge. So they are moving towards the electric field, which is actually anode, right? Right. So what happens is once the smaller fragments are moving forward when compared to the larger or bigger fragments. So that's what you are observing. Coming to the next slide. So once the separation is done here, the separated DNA fragments so will be visible only after they have been stained. So for that, we actually use a ethidium bromide, which is used as a compound like which normally strains the DNA, and later it will be exposed to the UV radiation. Okay. So you cannot see the pure DNA fragments in the visible light without stain. That's why we normally strain them. And after once it is stained, then we will expose it to the UV radiation. So in the UV radiations, we will chuck the bands in a proper way. That is, we can see a brighter orange color bands of the DNA and the ethidium bromide stain when they are being exposed under the UV light or to the UV light. And then the separated bands of the DNA are being cut off from the agarose gel and extracted from the gel piece. And this step is actually called as illusion. So that is you will choose a specific DNA which you requires and you are gonna cut that specific part of that specific part. Okay, and like that agarose gel, and then from there you'll extract that DNA. So the DNA fragments will later on be subjected to purify in this way. They are being used in the constructing what recombinant DNA by joining them with the cloning vector. So now we got a recombinant DNA or a desired DNA, and that will be constructed as a recombinant DNA. So by joining it with the one of the suitable vector. So like what vectors? And I think with cloning vectors, so where you can join your desired DNA to it. So here, vectors like plasmids, then we have bacteriophage. So they have the ability to replicate within the bacterial cell independently of the control of the chromosomal DNA. So vectors are present, they are engineered in such a way that they help easily linking of the foreign DNA like whatever the desired DNA that we put, right? So the link easily and selections of the recombinants from the non-recombinants. That means the selection of the recombinant desired DNA from non-desired DNA, something like that. So when I'm talking about a vector here, so we have certain places like where it has its own role. So one thing we do see in a vector is ORI, that is origin of replication, as a part or a place in the vector where the replication normally starts. So sequence where the DNA replication starts, any DNA linked to this sequence replicates in the host cell, controls copy number of linked DNA. Vector with high copy number, that is ORI, are referred for recognizing many copy of a targeted DNA. Then we will have a selectable marker. So selectable marker is used to identify and eliminate non-transformable, allowing specific growth of the transformants. Then so common markers that we use is gene providing antibiotic resistance, such as amphyllin and tetracycline. So these are the common markers, gene markers which we use. And coming to its transformation, so that is, it actually induces the DNA into the host bacterium. Then we have the cloning site. So vector should have a single recognition site for restriction enzymes to simply clone that. Okay. For example, if we have PBR, this is PBR 322 vector, which has a restriction site with an antibiotic resistance genes, that is BAM-HI site and tetracycline resistant gene. For tetracycline resistant gene, it will be like this is actually a cloning site. Then recombinants lowers resistance to one antibiotic drug to the foreign DNA inserting, but can be identified using dual antibiotic resistant test or chromogenic, substances. So chromogenic substances like beta galactosidase system. Anyhow, recombinants, no color produced, and where it comes to non-recombinants, blue color colonies you will see. So vector for cloning in plants and animals. Normally for plants, we use agrobacterium, that is tumor filicin, that is the tumor inducing TI plasmid, modified into non-pathogenic. That means which the tumor inducing plasmid, which is later on modified as non-pathogenic vector for delivery of the gene of the interest. Whereas in animal, we have ectoviruses, right? So they are also a pathogenic, but they're being transferred as non-pathogenic, modified as non-pathogenic, and used as vector to deliver desired gene. So these vectors, exploit the natural gene delivery mechanism of bacteria and virus for transformation in the eukaryotic host. So next we have is uh, micro injection of the DNA into the nucleus is another method. So where you will take the desired DNA now. So you have a DNA. So will be injected directly into the nucleus of the host. Okay, this is a cytoplasm host cell. So nucleus. In that way also we can actually bring out the desired DNA into the host. This is another method. So there is a process of recombinant DNA technology. One is isolation of the genetic material. So DNA genetic material has been isolated in most organisms. DNA is the genetic material. So where we actually use the purification step. So where breaking the cell. So first thing is we will take up the cell. Enzymes like lysozymes in bacteria. If it is plant, there's cellulose. Then if it is in fungi, chitin is used. So the cell wall, they are being made to break the cell wall. Then the RNA is removed with ribonucleus. Then the proteins are removed with the protease enzymes. Then other molecules are removed with a specific treatment. Then at the end, you're gonna add the chilled ethanol, which in addition to purify the DNA solution, causing DNA to precipitate as a thin thread. So later on, you'll take up that with the help of the restriction enzyme, you'll cut the DNA at a specific sequence, and then they are being incubated with the enzymes under optimum condition. Then agarose gel electrophoresis is actually used to monitor the digestion. Then DNA being negative charge move towards the positive electrode, that is anode. Then the source DNA and the vector DNA are cut into the same restriction enzymes, and the gene of interest from that DNA has been incorporated into the cut vector DNA. Then the DNA ligase is added to join the DNA fragments and forming what? A recombinant DNA, because host DNA is also there, then the foreign DNA is also there. So the next we have is amplification of gene of interest using the PCR. So one thing is we have a gene of interest here. So we have the DNA. So now the double stranded DNA, so it has been denaturated, okay, with the help of a huge temperature. Then once it has been denaturated, so with the help of the primers, so like these recombinant DNA will become separate into individual DNA, so a single stranded DNA. So once they become single stranded DNA, right? So primers are been added. So primers are added. These primers will go and recognize its binding site, right? So this is actually called as annealing process. So once that has been added, DNA polymerase, that is stack polymerase plus deoxynucleotides are been added. So what is the major role of them are it helps in the extension. Okay. So DNA primers will go and bind to its origin, like recognized site. So next DNA polymerase is there. Then we have that, then the deoxynucleotides are there. So what they normally do is deoxynucleotides, they actually produce the nucleotides, and as a reason, extension and DNA polymerase helps in extension. So that process is actually called as extension. So once it is done, now you will find the desired DNA now, right? So that will be amplified for 30 cycles. So at the end, you will have like 1 billion times of, uh, how do you say that? The desired DNA you will get. So one thing is inserting recombinant DNA. So whatever the recombinant DNA are there are introduced into the host cell using various method and making the cell competent. Like recipient cells are made competent to take up the DNA from the surrounding. Then transformation, like recombinant DNA with an ampicillin restriction gene is introduced into the E. coli. Then they have been transferred. The transferred cell gains resistance to the amplicin. Then how do you select? So transferred cells are separated as agar plate containing the amplicin. So transferred cells will obviously grow because now they have shown the resistance towards the ampicil. So in the agar plate you have ampicil. So since the only certain, like the one which is showing E. coli cells, which are actually showing this resistance will only grow. Then while the rest other E. coli cells which has no resistance will die. Then the amplicin and resistance gene act as a selectable marker to identify the transformed cell. So what is the purpose of the recombinant DNA here? Insert foreign DNA into the host cell to multiply it and produce the desirable proteins. Then gene expression, foreign gene has to be expressed under specific condition to produce the target protein. Then large scale production is also there, essential for obtaining sufficient quantities of the desired protein and requires optimization of the expression and large scale culturing method. Then we have culturing method like small scale culture method suitable for lab experiment, yield limited itself, where continuous culture method, fresh medium is continuously added and use a medium removed to maintain the cell in an active phase for higher protein yield. So what is the limitation of small culture here? So it cannot produce large scale, more yield. So produce for large scale needs. So then we have bioreactors. So bioreactors are one where a large vessels designed to process large culture volume. So convert the raw materials into specific products like proteins and enzymes. So here the bioreactors mainly has an ideal conditions including its temperature, pH, nutrient value and oxygen. Then the major purpose is to produce the large scale desired biological products. So what all it consists? One is stirred tank reactor. It is actually a cylindrical or curved based design to ensure the effective mixture. So stirring, it actually ensures even mixing and uniform oxygen distribution. And alternatively, air can be bubbled through the reactions of oxygen supply. So what all it consists? One is agitatory system that is ensures a proper mix, right? Then we have the oxygen delivery system, which maintains what? An oxygen level. Then foam control system, which prevents excessive foam formation. Temperature control system, which maintains an optimum temperature. pH control system, which regulates the alkalinity and acidity. And sampling pods allows the periodic withdrawal of the small culture sample. Then we have downstream process, which involves the separation and purification of the product after the biosynthesis stage. Then the purified product is formulated with the suitable preservative. Then clinical trials will be done, like for products like drug. The formulation undergoes thorough clinical traits and strict control quality control testing is required for each product. The specificity of downstream processing and testing depends on the products and its type. So this is all about the biotechnology and its processing. Even it could be in the small scale as well as in the large scale. So this is all about the chapter. If you feel any doubt, you can contact me through the comment section or else the Telegram channel. Don't forget to like, subscribe, and also share with your friends. Thank you.